All right, in the last section, we talked about muscles and joints, just kind of generally speaking in terms of the muscles. This time, we're going to talk about how muscle contraction actually works. What the muscle cell basically relies on are these microscopic little structures that are going to kind of work, with one another to basically shorten the muscle. So the contraction of the muscle is actually the shortening of the muscle. And you've got a variety of these microfilaments that are, kind of contained within this structure here. And the structural component of the muscle cell is known as the sarcomere. Okay, and then you'll see that we have these Z lines right here, and those Z lines come from the actin fibers. We have the M line right here, and that's what's made up of the myosin. And this movement basically is governed by these chemical energy transformations. In other words, the hydrolysis of ATP. As this ATP gets broken down into ADP and PI, and we release a little bit of energy, that energy is used to shorten the muscles. Okay, so as far as the skeletal muscle is concerned, and it's also referred to as striated muscles, and just recall that it's one of the three main types of muscle fibers that we've learned about. We've learned about cardiac muscle, we learned about smooth muscle, and this is the skeletal muscle. And this attaches to the bones, of course, we said, and provides for the movement. And it's kind of a hierarchical scheme of smaller and smaller subunits. So you can see we've got the muscle, for instance, right here, and then we've got a bundle of muscle fibers that makes up the muscle, and then that bundle of fibers is kind of like a collection of these single muscle fibers, and then the muscle fiber, these are going to consist of these myofibrils right here, and then still further, as you zoom in, you've got the sarcomere, okay, and that's the actual structural unit of the muscle. And the skeletal muscle usually is a bunch of, or a single cell rather, with a bunch of different types of nuclei in them, or a different nuclei. It's a multinucleated type of cell, and it's going to carry out the various functions of the cell, which is movement largely, okay. So these small units called myofibrils consist of the thin and thick filaments, okay? And the thin filaments now, the thin filaments are the actin that we said, okay? So these yellow things right here, these are the thin filaments. These things right here and so forth, these are the thin filaments, and they're consisting of actin. And then they've got two strands, as you'll see, you can't really see in this picture, of tropomyosin, which is kind of coiled around them. And we'll kind of get into more of the details as we move through here. And then the thick filaments, these are the myosin filaments, okay? So that's what we're seeing right here. And, these thick purple things, those are the myosin fibers. And then the sarcomere, this is the contractile unit of the actual cell, okay? And each one borders with another one. So in other ones, you can see kind of a small component of this one here. These would be extending out, and then they would attach to another actin filament, and then you would see that you've got the, or excuse me, let me undo a couple of these things here. These myosin fibers, okay, these thick filaments, these are attached, and then these would be attached to another M line here, which is going to be attached out. So these things are basically all adjacent to one another, okay, if we're looking at this here. And so they're all along the muscle fiber. You can see here's a sarcomere right here, okay, that's bordering up with another one, which is right here, which is bordering up with another one, which is right there, and so forth. So they're all along the length of the muscle fiber, okay? And you have the M line, you have the Z line. The M line is made up of the myosin. These are the thick filaments. The Z line is the thin filaments, and those are the actin fibers. And then at rest now, the relaxed muscle, you can see here, there's a lot of open space, for instance, right here and right here. And then as we look at the electron micrograph, we've got the TEM of the muscle cell here. You can see all through here, there's a lot of open space as well. And it's important to keep in mind that when the contraction occurs, the actual length of the myosin fibers and the actual length of the actin fibers is not going to change. They're just going to slide. So my fingers aren't getting any shorter, but my hands are getting closer and closer together. And that's kind of the way this is going to work as far as the overlap occurs. So you can see here that when the myosin and the actin are interacting, and we'll talk about how here in a sec. When these things are interacting, they're pulling one another. The myosin is pulling the actin closer together. So you can see during contraction now, these Z lines are getting closer together down here during contraction. Okay, so it's important to keep that in mind. And when you look at the electron micrograph right here, you can see that this structure is much shorter than that structure, and that's what's happened during contraction. Okay, is the Z lines are actually getting closer together. Okay, so it's important again to note that the thick and the thin filaments are not changing length. They're just kind of sliding over one another, overlapping more, and pulling those Z lines closer together. Okay, so now as far as when contraction actually occurs, we have the hydrolysis of ATP. Okay, so when ATP, okay, just recall this from earlier in the year, when this gets hydrolyzed, we get ADP plus PI plus some energy. Okay, and that energy now is going to be used to kind of pull those filaments kind of over one another and contract the muscle. And the myosin head now, which is if we go back, for instance, we can't see right here, but all along these little purple structures here, all along those structures are numerous myosin heads. So we can kind of see them right here, all along here. That's what these things are. And these myosin heads now, when they hydrolyze ATP, they get kind of moved into a high energy configuration. So this is the low energy configuration right here. And then when ATP gets hydrolyzed, it moves into a high energy kind of state. And now it has the ability to attach to the actin filament and pull it, pull those Z lines closer together and shorten the muscle, contract the muscle, and provide for some movement. Okay, so we can see as we go through here, here we have the low energy state. Okay, we hydrolyze the ATP, get it into a higher state. It binds to the actin, and then it moves or slides the actin in this direction here. Okay, this is going to get moved. Okay, this yellow structure here is going to get moved in that direction. As it pulls, then it lets go, and the cycle repeats, and it does very, very rapidly. This occurs, which is what's providing for the movement. Okay, so now as far as if we look a little bit closer at the actin filaments here, you can't really see the tropomyosin. But here when we look, here's this, this is the tropomyosin that we've been talking about, and that's that gray kind of cable that's wrapped over the entire actin filament. And what it's doing is it's blocking the myosin binding sites. So when we back up here, you can see that the myosin is going to bind, for instance, once it gets into the high energy state, and then it cross-links or binds to that actin. Well, the myosin binding sites are blocked here, and then when we release calcium, and we'll put all of this together in a manner that you can understand in a second. When we have calcium present, the calcium binds to the troponin complex, this purple thing here, causing the tropomyosin to slide, basically, to move. The whole things are going to move, and then you can see all of these myosin binding sites start to get opened up, and that's what they're going to grab a hold of when they're contracting. Okay, when they're pulling the actin together. Okay, so at rest, you can see the tropomyosin covers the binding sites, and those actin filaments are unable to bind with myosin and provide for movement. But once the calcium starts to accumulate in the cytosol, now we're going to have the myosin binding to the actin filaments and result in some contraction here. Okay, so you can see this is what we just went over. Without, it doesn't have the tropomyosin, but all of this is going to occur when calcium is present and the tropomyosin has slid out of the way, making the myosin binding sites available. Okay, so now what we want to do is we want to take and kind of incorporate some of the stuff we learned about action potentials into the resulting muscle contraction, because we said that the axons can synapse with another muscle cell, or another neuron, or they can synapse with a muscle cell. So in this particular case, what we're going to be talking about is how the synapse kind of coordinates muscle contraction. So we get these signals here from the motor neurons, okay? And a motor neuron is anything that's going to provide for movement. A sensory neuron is going to sense the stimuli from the surroundings. But the motor neurons now, these are going to stimulate the release of the calcium ions from the sarcoplasmic reticulum into the cytosol of the muscle cells. And that increase in calcium, as we've seen, is going to what's going to result in the contraction of the muscle. Okay, so the first step in the muscle contraction is the arrival of an action potential at the muscle cell through a motor neuron. Okay, and this action potential is going to stimulate the release of acetylcholine. So you can see here all along these muscle fibers, we've got these nerve cells that are kind of synapsing with the muscles. Okay, so what happens is now that action potential is going to lead to a depolarization of the plasma membrane. That's going to contribute to an action potential within the actual muscle cell itself. And the action potential is going to travel through these structures in the muscle called the transverse or the T-tubules, okay? And that spreads towards the interior of the muscle. And you can see now here with this picture, this is the sarcomere that we were talking about. Here it's actually covered with this blue thing, and that blue thing is the sarcoplasmic reticulum. That's what holds the calcium, all right? So now, if we look at this, we get an action potential that comes down, okay, depolarizes the membrane, and we get the release of the neurotransmitters. Just like we said, it's going to result in calcium coming into the neuron. The neuron is going to, that's going to stimulate the release of the neurotransmitter from these vesicles. The neurotransmitter in this case is acetylcholine, and then that is going to depolarize the membrane of the muscle cell, and that action potential now is going to spread down the muscle cell to the T-tubule, where when it travels down the T-tubule, it's going to cause calcium to be released from this sarcoplasmic reticulum, this blue structure. As the calcium now gets dumped into the cytosol, it has the ability to bind with the troponin. And when it binds with the troponin, remember we said earlier that that causes the tropomyosin to slide out of place and open up the myosin binding site, enabling the myosin heads to bind to the actin and shorten the muscle fiber, resulting in muscle contraction. Okay, so these are some of the words that are just describing what we were saying. Again, kind of the opposite here. When we don't get an action potential, now the muscle is going to relax. We don't have any neural input. The sarcoplasmic reticulum is going to take up the calcium from the surroundings. The calcium is going to not be bound to the troponin, and that tropomyosin is going to slide back into place, and now the muscle is going to be ready for the next round of contraction. And all of this occurs very, very rapidly and results in the movements that we, do on a daily basis all the time. All right, so if you have any questions, make sure you write them down. There's a lot kind of to this, but it's mostly based in the vocabulary. If you understand the vocabulary, then you can kind of put together what you know. And then when you know how the muscle contracts and you can put it together with the vocabulary, then I think you'll be all set. All right, so if you have any questions, write them down. We'll talk to you soon.